Okay, so we're at the last organic molecule that really comprises important pieces of our body, or important structures in our body, and that would be the nucleic acids. So there are two major types of nucleic acid: DNA, which you've probably heard about, and that stands for deoxyribonucleic acid, and RNA, which stands for ribonucleic acid. DNA and RNA are both made of chains of these small little units called nucleotides. And they join together in chains very similar to how amino acids do when they form proteins. So all nucleotides have three components to them, and you're looking at a nucleotide here. One component is a 5-carbon sugar, and so all that means is where you see these points in this pentagram, that's where a carbon would be located in that little molecule. So a 5-carbon sugar. They also have a phosphate attached to the sugar, and then they also have another component attached to the sugar, and that is called the base. Sometimes it will be called the nitrogen base. Sometimes it's referred to as the nitrogenous base. I'm fine if you just call it the base. That's what I usually refer to it as. So this is a nucleotide. This base can be one of 4 bases if this is part of a DNA molecule. And if it's part of an RNA molecule, it also will be one of 4 bases. And we'll talk more about the types of bases that are involved in each of these different molecules. So DNA stands, as I said, for deoxyribonucleic acid. DNA contains instructions for making nearly every protein that our body needs. So the instructions are kind of contained within the specific sequence of nucleotides in the DNA. So a certain sequence of DNA, called a gene, carries instruction within that sequence of different nucleotides joined together that will end up eventually making a protein. So we have many, many genes because we have many, many proteins that need to be made. All right, let's see. So in DNA, this 5-carbon sugar is called deoxyribose. And the base that you see here attached to that is the base adenine. That's shown to you right here, adenine. There are 3 other bases that could be attached to a nucleotide in DNA. It could be cytosine, thymine, or guanine. So those are the 4 bases that you find attached to nucleotides in DNA. So it takes many, many, many nucleotides joined one to the other to make a strand of DNA. And the DNA that's in our cells is actually a double strand. So you have 2 strands that are bound together, kind of like this, bound together. And I'll show that to you in just a second. Let's just take a look here. So this is kind of a busy picture, but here you're seeing that example of the nucleotide. And you notice that this nucleotide is shown to you right here. You also notice that there's a bond that exists between the sugar of this nucleotide and the phosphate of the next nucleotide. And then the sugar of that nucleotide is bound to the phosphate of the next nucleotide. So these nucleotides join one to another between their sugars and their phosphates. And we say that in this one strand that you're seeing here, this strand has a sugar phosphate backbone. Okay? And coming off of that backbone, you have these bases, right? Well, as I said, DNA is a double-stranded molecule in human cells. And so this is the second strand here. There's your phosphate sugar backbone, and here are the bases. Well, these strands have to link to one another, otherwise they'd just be all loosey-goosey. And they do bind to each other between the bases. So the sugar phosphate backbone is on the outside, and the bases point inward and bind to each other, and they form these little hydrogen bonds between them. Okay? And once that has happened, the whole molecule will twist into this helix shape, like you see here. And that is the form of DNA in our cells. So DNA nucleotides bind in a very specific way. A's can only bind to T's, and G's can only bind to C's. It's the only way that it can happen. One molecule of DNA in humans contains more than 1 million base pairs. So that's a base pair, that's a base pair. More than 1 million in just one molecule. That's insane. Yeah. And these individual nucleotides in each chain are in very specific sequences, as I said. And whatever the sequence is, that's a gene, and that will determine what protein ends up getting made from that DNA sequence. So, although every person's DNA is different from one another's, the DNA in every one of our cells within a single human body is exactly the same. The one exception is with identical twins. So identical twins, they have the same DNA sequence in every single one of their cells, but theirs is also identical to their identical twin. So that's the one exception. All right, so we say that the DNA molecules carry blueprints for traits that get passed down from one generation to the next, and that is called heredity. The other type of molecule where nucleic acids are used in, at least in another big one anyway, is RNA, which again is ribonucleic acid. It plays a very important role in making proteins. So there's a few different types of ribonucleic acid that help to make proteins, and we'll get into that in another chapter. But I wanted to cover a little bit about RNA. So just like with DNA, RNA is made of these nucleotides, just like you saw before. But the difference is that the sugar in RNA is called ribose sugar, not deoxyribose. It's still a 5-sided sugar, but just a little different. It also has a phosphate group, and it also has a base coming off of it. But the bases in RNA are similar but different. So 3 of the bases that you find in RNA are identical to the ones that you find in DNA: adenine, guanine, and cytosine. Uracil is different. In DNA, the base that would be here is thymine, but in RNA, instead of thymine, it has uracil. They're very close in structure, but just different enough to be different. So some RNA molecules are temporary copies of DNA genes, and we'll talk about those in more detail later, but we call those messenger RNA molecules. And some types of RNA molecules actually latch onto the amino acids and bring them over to where proteins are being made to deliver the amino acids so they can get added to the chain of growing amino acids when a protein is being made. And we call those transfer RNAs, or tRNA. And you're looking at a transfer RNA molecule diagram here. So there are other functions of nucleotides. One very important molecule that contains a nucleotide is called adenosine triphosphate. And we shorten that and just call it ATP. So adenosine triphosphate contains the nucleotide adenine. And adenine, here, that you're looking at here, has a 5-sided ribose sugar, and it is attached to, the adenine is attached to the ribose sugar, forming what is called adenosine. So the adenine base plus the ribose sugar makes a molecule called adenosine. And attached to adenosine, you have 3 phosphates. The bonds that exist between these phosphates hold a ton of energy. And every cell in our body uses this molecule, this ATP molecule, when it needs energy. So what happens is, when a cell needs ATP, one of these phosphate groups is broken off from the ATP. And when that happens, as you know, when bonds break, they release energy. And this energy gets captured by the cell in order to allow a cell to do some kind of process. The remainder of this molecule is now called adenosine diphosphate because it only has 2 phosphates. And this molecule gets recycled. Some energy from some nutrient catabolism is going to allow a phosphate to get put back onto that molecule, forming ATP. And then when the cell needs energy again, it'll pop the phosphate off, gather the energy that is released, and the cycle will continue and continue. Lastly, I just wanted to go over names of molecules and what they can tell you. So you may find this helpful or not, but when you're looking at a name of a molecule like adenosine triphosphate, the base word of the base of the word, I should say, is usually telling you that that is the largest component of the entire molecule. So even though these are 2 words, the base word is triphosphate. Adenosine is used kind of as a prefix. So what that means is the largest part of adenosine triphosphate are the phosphates, and it has a small adenosine group attached. The term lipoprotein is a protein with a lipid attached. So it's primarily a protein, but it has this lipid attached to it. And a glycoprotein, same thing. It's primarily a protein with a sugar attached to it. You might also see something called a proteoglycan. A proteoglycan is a sugar that has a protein attached to it. So hopefully you kind of can follow what I just went through. Read this over and please let me know. I think this is the end of our lecture. Ooh, I bet you're happy. No more recordings. But please take your time with this. Take good notes, and you can email me topics that you're going to want to discuss when we meet on Monday at our virtual meeting. That would be helpful to me. You don't have to do it, though. That's just kind of gives me a heads up of what I want to make sure I cover. But you can just come to the virtual meeting and ask me questions as we're working through stuff. So anyway, you know how to reach me by phone, text, and email. So I'll be talking with you all soon. Happy studying.